Thanks everybody for attending. This is the annual research retreat of the Department of Orthopedics. And this year's retreat is actually a co-production between the department and REACH. REACH is the acronym for a very complicated name that is basically our NIH-funded research, low back pain research center here at UCSF. For those of you that aren't familiar with HEAL, HEAL is, it stands for Helping End Addiction Long Term, and it's the NIH's response to the opioid crisis. Many patients that end up addicted to opioids actually start out as low back pain patients. So HEAL is, NIH, I should say, has taken a real interest in low back pain research and has devoted something like $500 million to that effort. So we were very fortunate here in REACH at UCSF, I should say, to get funding to establish REACH. I should also mention that there's a number of other research groups here at UCSF that receive funding, including Sharmila Majumdar, who's one of our speakers today, and Aaron Fields and Prasad Shervalkar, who's in the pain management department and collaborates with the neurosurgeons. So our mission in REACH is to really bring precision medicine to low back pain care. And as someone who spent over 30 years trying to figure out why back pain patients hurt and what to do about it, it's a very tall order. I've been down a lot of rabbit holes over the many years and had pretty much given up hope before REACH came along, but I'm super excited about what we might be able to accomplish, and very excited about hearing from this great faculty we have today about how we can really bring that vision, precision medicine for low back pain to reality. REACH is hosted by the department, but it's a multidisciplinary, multi-institutional program. We have collaborators across the country, actually, and even as far away as Finland and Switzerland. I think in a busy clinical department like ours, it's easy for researchers and clinicians to sort of get into their silos. And one of the great things about REACH has been to see for lack of a better term, the magic that can occur when clinicians and scientists really focus together on a common problem. And that's been a great experience for me, and I think for everybody else in the program. One of the reasons we're having this joint retreat is we want to try and figure out ways that we can leverage our experiences in REACH to improve research in the department, and that comes with developing tools, of course, but I think also just as important is figuring out how we can sort of create that same sort of collaborative energy and bring it to other research problems in orthopedics and musculoskeletal care. We have a great program today. We have a number of people to help put it together, including Jeff Lotz, who's the PI for REACH and provided the spark for this. And of course, great kudos to Jeff for getting this whole effort going here at UCSF. Jeannie Bailey and Irina Strigo are researchers. Jeannie's in the department where she focuses on biomechanics, and Irina is a pain researcher at the VA. And her and her colleague Wolf Mailing, who's at the Osher Center, have formed our what we call our biobehavioral core, and they're a great example of what clinicians and scientists can do when they put their heads together. Other important members of the team that put this retreat together are Patricia Zhang and Peter Wu who are my colleagues in the physiatry section of the department. And also Rochelle Polkowski, who directs our clinical research efforts, and of course Tom Peterson, who's both in our department and the Baker Computational Institute. So with all of that, I think Tad, our department chair, is going to say a few words before we get kicked off with our program. So I'll turn it over to Tad.
Thank you, Connor, and it will be a very few words, but I really want to recognize and say thank you, Connor, to you, to Jeff, the PI, and all the individuals that you mentioned for achieving the REACH grant, and then creating an environment of opportunity that has been spun off of the activities in REACH. And as Connor alluded to, one of my great hopes for this gathering today is that it's a collection of people who are participating in REACH and others who are coming from other disciplines. But my hope is that not only will this activity support what is happening and expand and leverage what is happening in REACH within the focus topics in REACH, but also bleed out into other disciplines where some of us might look at what's happening in REACH, what applications of technology and resources and research questions are being asked, and how some of that might be applied into other disciplines, other musculoskeletal disciplines or beyond. I think that's an opportunity that really expands the impact that REACH can have in our department and at the university and beyond, with collaborators outside the university. So very appreciative of all of you who have committed the time and attention to this. Encourage, if you're able to turn your screens on and be fully engaged with the speakers. I think it's nice for the speakers to see an audience out there. And thanks for this opportunity. I look forward to seeing how the morning unfolds and what comes after that. So Connor, appreciate the opportunity to say a few words and look forward to what follows.
Great, thanks, Tad. So before we get started, just a couple things. Jake, do you want to we don't have to show a slide, but just mention real quick what the format is for questions, or you can show a slide, that's fine.
Good morning. Yeah, I just wanted to mention to everyone, so when it's time for questions after each of the presentations, if you have a question, please raise your hand in Zoom and we'll call on you to unmute yourself to ask the question out loud. And then just one more reminder, we posted this virtual program link. You should have received a link, but it's also in the chat box. So just a reminder that each of the breakouts is a separate Zoom link, which is in the program, but the REACH hypothesis breakout is actually this same main Zoom link, but you can always refer to the program to find those Zoom links. And if you have any difficulties during the day, please reach out to myself or Daniel Davis. Thanks.
Great, thanks, Jake. And I want to echo Tad's request to turn your video on because it's really, really helpful when you're giving a presentation to see people's faces out there. So is Keith is here, I hope?
I'm here.
Okay,
He is.
So I'm going to introduce our keynote speaker, Keith Yamamoto. And Keith has really been a visionary researcher and leader at UCSF for decades now. And you can just refer to his bio in the program to get a complete listing of all his accomplishments. Keith was really there at the birth of precision medicine. And he now directs the Precision Medicine Center at UCSF. And for those of you that are interested in precision medicine, I encourage you to look at their website. It's got a lot of great information about the different initiatives here at UCSF. It also has a great picture of Keith shaking the hand of Barack Obama in the White House. And if you ever take a look at that, you'll see a very happy Keith smiling at Barack Obama. It's a wonderful picture, so check that out. So Keith, thanks again for taking the time to talk to us, and really look forward to your talk.
Thank you, Connor. Let me share my screen. But thank you very kindly for the introduction and the invitation, opportunity to join you all today. Whoa, that's not where I wanted. That's most of the way through my presentation. Let me stop that for a second and get to the right screen. There we go. Let's try that one. How's that? Okay. So thank you for the invitation. And let me start with a kind of a shout out to your department, orthopedic surgery. Over the years, I've received great advice and counsel and treatment from Department of Orthopedic Surgery, notably Connor himself. Actually, Connor, it's great to see you in this context rather than in the clinic, but anyway, it's good to see you. From Sig Bervin, from Ben Ma, and from your chair, Tad. And in the last 15 years, I've had 2 shoulder surgeries and 4 spine surgeries. So I'm a repeat customer and a very happy one. So thank you for all of that attention. It's great to be able to tap into the UCSF clinical enterprise in such a way that has been successful and happy. So as I stared at this title that was assigned to me, it brought to mind 2 papers that were published in 2018 that I think are notable. One is from a group of investigators at Bentley University in Massachusetts, where they just compiled data and pointed out that between 2010 and 2016, 7-year period, 210 new molecular entities, drug candidates, were approved by the FDA. 210, 30 a year for 7 years. And what was striking about that is that they tracked back to see what the source of support was for the investigations that led to those FDA-approved drugs. And for all 210, the work began with NIH-funded research, and almost all of that was NIH-funded basic research. Just that made me happy. I'm a basic researcher, so that's good. And the second paper was authored by Mark Fishman and his colleagues at Harvard Medical School, and published in Science Translational Medicine. And what they did was they looked at what had been voted the 28 most impactful drugs on the market. All right, this is a physician survey. That doesn't matter, but they took they just took that list and asked what was the originating work that led to these drugs that are now so impactful, affecting so many people, the health of so many people. And what they found was that 24 of them, 85% of those, came from basic research, fundamental, untargeted, curiosity-driven science. So that, again, that's good and makes me happy. But the important piece of data was that the time gap between that initial discovery and the FDA approval of the new drug that became so impactful averaged 32 years. Really remarkable. Then if they took the 12 of those 24, half of the 24 that had been approved the earliest, and weighed them against the 12 that had been approved most recently, they found that the earliest ones, the time gap was 36 years, and the later, more recent ones, 26 years. So it's getting better, but 26 years is sort of a long time. And it reminded me of my first R01 grant in 1976, I might add. And we've studied throughout my career the molecular biology of transcriptional regulation in eukaryotic cells, in worms and flies and yeast and mammalian cells and mammals. So basic science. And in 1976, as now, in the abstract of the application, we were compelled to write something about how the work we're proposing relates to health and medicine. And I can assure you that in 1976, we were just making it up. We saw no link between the kinds of stuff that we were doing and health and medicine. So we just made stuff up. Maybe it'll apply to cancer, and went on our way. And the NIH was happy with that, and we got the grant, and things went on. But really, what these two stories tell you about those papers and about my grant application and making up the health relevance is that the connection, the link, the communication between the basic science and the translational and clinical science was not good. All right. Yes, it's improved. It's gotten a decade better from 36 to 26 years, but still needed work. All right. So that was kind of, in a way, what really got me started thinking about this notion that turned into precision medicine. Now let's leap ahead to 2009, and I am I've become the chair of the Board on Life Sciences for the National Academies, and we the board decides to assemble a consensus report committee. This is a committee of distinguished scientists that is going to write a report on a specific topic that we lay out. And I was able to convince our then Chancellor, Sue Desmond-Hellmann, to co-chair that committee. I put myself on the committee because I was excited about the potential for the work. And the challenge to the committee was to consider whether what the wisdom would be for building a research and health continuum, really bringing together these disciplines and separate communities to interact much more effectively and freely. And the committee said, gee, if you could do that, it would be a good idea, because if you could really integrate the rapidly expanding range and detail of biological, clinical, behavioral, experiential information, social determinants, then you would have a chance to drive basic discovery with new syntheses and hypotheses that the basic scientists wouldn't think of if they were still just stuck in their little domains. And it would create a new classification of disease based on mechanisms instead of organs and symptoms. So you would be looking at the connection to disease all the way back to what the causative components were, and there's establishing that continuum. And this in turn would advance the speed and accuracy of prevention, diagnosis, treatment, and therapy. Great aspiration. How do you do it? So the committee went on to say, you do it by building an enabling tool, building basically data, building information commons and a knowledge network, and I'll talk about both of those, to correlate and align this disparate information. By bringing the information together, it would bring researchers together, and motivate team building, and convene all the stakeholders, including patients, into this new dynamic research ecosystem. So that's what this National Academies report that was published in 2011 said, and you can find that report. I think it's an interesting read. So it took us 2 years to finish this report. We started in 2009, published it in 2011. And here I am at the last meeting of our committee, which I hosted at UCSF. So we were up in the stem cell building conference room. And here I am. You can see it's 10 years ago because I have long hair and a BlackBerry, which I still miss. And I'm taking a picture of the whiteboard after the committee brainstormed about what they were going to call this report. And toward precision medicine, a new taxonomy of disease, a knowledge network, and a new taxonomy of disease. So that's what happened in 2011 that published. Since then, the visions of precision medicine, as the slide says, have been continuously built out. As I had hoped when I convinced Sue to co-chair the meeting, she came back to UCSF and in 2012 declared precision medicine one of the primary organizing principles for the way that UCSF would go forward in the future. We continue to work in the White House to convince President Obama that this idea had merit, and a presidential initiative with funding, but the really push of the president behind it would be impactful, and he declared that in 2015. And we worked in Sacramento to convince Jerry Brown, Governor Brown, that he should launch the California Initiative to Advance Precision Medicine, and he did that. And I'm still involved in both of these endeavors. I'm actually a vice chair of the California Initiative to Advance Precision Medicine Advisory Council. We just met yesterday. So things have gone forward. So precision medicine then is this approach to research health and healthcare that harnesses vast amounts of data and health information from basic clinical and population health studies to define biological processes and disease mechanisms, and to reach toward individual, predictive, and preventative care, individual care, precision medicine down to the level of the individual. And as I said, this is really central to the UCSF institutional vision, mission, and operations to conceptualize and build precision medicine by capitalizing on how amazing our community is, to motivate reaching out to partners, convening stakeholders, and creating this matrix that yields insights into the determinants of health and disease. One of the aspirational goals of precision medicine was nothing less than to transform healthcare. And so, these are a little glib, but just stay with me here. So current practice, at least in glib language, is to use the vital signs that are measured today, that is the day that the patient walks into your clinic, to compare them to the vital signs at the last visit. Maybe if it's a fancier place, you have more information on the EHR. Certainly UCSF we do. To assess those the symptoms that made the patient show up, and then the physician uses her expertise and experience and judgment to reach a diagnosis and a treatment plan. Don't let me be too critical of that. It works. It's terrific. But in fact, this notion of personalized medicine says we can do better by collecting more data, more information about the patient, patient's electronic health record, of course, and extending all the way to more refined stuff like the human genome. And then the physician uses all of that information to make a more informed diagnosis and treatment plan than she would have made without those data. Precision medicine then takes it another step further and says, use the computational knowledge network that aggregates and analyzes information from across not only patient cohorts and healthy populations of people, but experimental systems where a lot of the fundamental processes, biological processes have been defined, and the things that go wrong with those processes that lead to disease have been uncovered. And then really reach toward precision prevention, diagnosis, and treatment at the level of individuals. So that's kind of the aspirational plan. What we've done at UCSF is to build basically a precision medicine platform machine, if you will, a dynamic, data-driven machine that cycles data and knowledge and new information iteratively. So it begins with the three ways that we all work: basic research, clinical discovery, population science. And those three foundation pillars of research then are actually carried out using a set of enabling tools, kind of ethics and engagement, kind of an overlay for everything that we do, hopefully, but also tools, omics, imaging, computational health, and increasingly digital health. And I'll say a word about that in a minute. And so this this usage of tools by the researchers of 3 different stripes then generates data, lots of it. And those data then go into one place, an information commons, a computational platform in which all of the different data types can be integrated so they can be compared and analyzed. That process of analysis uncovers correlations and patterns and connections for information between the layers, right? And that's what constitutes the knowledge network. And from that knowledge network then comes new knowledge, which then feeds back into the system, right? Generating new ideas for discovery or new ideas for technology to make the tools better. And the information then iterates and cycles back through. All of it to the benefit of people and all kinds of people, researchers, clinicians, patients, well people, and so forth. So this whole machine is set up to serve that population. So just to take a little tighter look at what is meant by this knowledge network idea, it's really just the principle is Google Maps. That is, separate into different layers, different data types, right? And in Google Maps, of course, everything is aligned by geography positioning. And so if you just drop a plumb line through any point on the grid here, you would then be able to ask, where are the Starbucks relative to the railroads, right? In the case of the information commons, you have all different kinds of data, but they're not aligned. They don't have any registration point. And so making connections between some observation in the exposome to some data, new observation in the epigenome requires that pesky research thing. And that's how those connections are made. And from those connections then is realized the knowledge network. So build the knowledge network that uncovers connections between information from basic clinical and population studies to make use of all discoveries in reaching conclusions about what happens with a patient or what the next hypothesis is for basic research. So this is really nothing more than a slightly refined conclusion of that National Academy's report to build an enabling tool. UCSF's knowledge network is here. And it's called SPOKE, Scalable Precision Medicine Oriented Knowledge Engine, SPOKE. And really what the cartoon shows is that it's a graph approach to bringing together data of different sorts that you can see diagrammed here. This is some of them. And then building connections between them. So right now, the Spoke Knowledge Network has more than 3 million nodes. In fact, this slide is a few months old. 3 million nodes, and then 5 million edges, connections between the data layers. So it's growing all the time, and getting stronger as more data are put in and connections are realized. Sorry about the truck going by. The process is very dynamic. In fact, the network is refreshed weekly, and then APIs are built to then connect to the knowledge network for specific purposes. And I've listed a few of those here. Repurposing drugs, supporting the molecular tumor board, finding drug targets, and so forth. Integrating the EHR is a big challenge that has been undertaken and has little pieces of completion elements in it, and to create a patient-specific health barcode that with thousands of interconnected variables, enabling tracking and prediction. For quite interesting process Sergio Berenzini has pioneered. And so I can just say that the COVID pandemic really revealed some real power to this network by connecting information about small molecules and symptoms of COVID in this case. The network was able actually to predict that dexamethasone would be an effective potential treatment for COVID-19. By connecting information about biological processes, about small molecules and biological pathways, Nevin Krogan assembled an incredible consortium that looked at the cellular proteins that the SARS-CoV-2 proteins interact with, and from that derived a potential repurposing drug repurposing list that identified molecules, many of which are now in clinical trial, to see if drugs that hit cell proteins that the viral proteins interact with block that interaction and thereby block the infection or the replication or uncoating or whatever it is that those proteins are involved in. Very exciting and innovative approach. So still to come, not on here, and really still to come, is crucial, and that's building social determinants of health into this network so we can tie those determinants to the kind of objective determinants that are listed here. So a lot still to be done, but a lot that has been done. Topical programs in precision medicine have been built and are expanding or launching or organizing in cancer, women's precision medicine, children's, public health, neuroscience and neurology, immunology, and others. Each of those programs encompasses all of the types of research that are done. Each of them benefits from and contributes to the precision medicine machine. So the more data that goes in, the more valuable that anybody's data becomes. There are, of course, gains from intersections with other programs, things that don't have to be rebuilt in a program because they've already been built in another program. And the whole process is used to identify, reach out to potential external partners and donors that can benefit the program. There are many precision medicine activities at UCSF. There's no way that I could list them, but here's just a few examples in specific domains. And I'll just call out one in each. I'm not going to show any data about them, but in the area of precision diagnosis, some of you probably know about the Siren and Gravity project programs that were launched by Laura Gottlieb and Nancy Adler. Mark Savage is principal in that as well. Really an effort to write code to develop data and exchange standards for social determinants of health. A big challenge, but something that this group is undertaking, I think, in a really exciting way. Digital health innovations. You're going to hear today, as soon as I shut up, from Riley about the bridge algorithm that they have built to connect precision medicine innovations for MS, for traumatic brain injury, for memory and aging, and other neurological disease programs to the basic sciences. Very exciting endeavor that she'll talk about. Precision public health is really the home where social determinants are being mapped out. They're connecting to the Siren and Gravity project and other social determinants programs, and an exciting endeavor. And just to call out the UC Data Warehouse that Atul Butte oversees for the whole UC network, that is building connections to the 17 or maybe more than that now, million electronic health records across the UC systems. So, so I've benefited from this invitation to begin learning about precision medicine activities that contribute to musculoskeletal care. And that's terrific. We'll hear from Sharmila about her marvelous work on machine learning and musculoskeletal imaging research. I've had the pleasure of learning about this in other contexts and interacting with Sharmila. Looking forward to getting an update. And then Connor was good enough to share with me information about these two exciting programs that are underway. The REACH project, which is identifying chronic lower back pain subtypes, defining patient phenotypes and underlying mechanisms and outcome preferences to guide with machine learning paradigms the chronic lower back pain treatments at the level of individual patients, precision medicine. And then finally, this new grant that was apparently just awarded called Expert Augmented Machine Learning for chronic lower back pain, really combining the clinical judgment of causes and mechanisms of chronic lower back pain and patient characteristics with machine learning driven clinical data mining. I think that's a really interesting proposal. I'm really excited to see how that comes out. And I'll be interested to see actually whether the expert judgment will be something that will always be essential as a second step to, no matter how sophisticated the machine learning gets to be, I suspect it will, but whether the causes and mechanisms of chronic lower back pain will become embedded in the machine learning side as the data become better and more sophisticated or systematized, and whether the outside experts that are imagined for this proposal will simply be dispensed with, and it'll just be the practitioner, him or herself, who then exerts the expert judgment overlaid onto whatever comes through the increasingly sophisticated machine learning stuff. So we'll see how that goes, and it'll be interesting. This is all terrific to learn about. I'm sure it's only a small part of what is being done. And I would just encourage these programs to build connections to the institution-wide knowledge network. I think that'll be to the benefit of both sides. So my awareness and interest in what's going on in your department and your programs is definitely peaked, and I look forward to the talks that follow. My last slide then is just to summarize what I think are really the impacts of precision medicine, what makes the program important. The knowledge network, as I've said, builds iteratively and contributes continuously. That is to say, it doesn't have to be done to have an impact. Whatever data is in there is going to help to build increased knowledge, and we just keep going. It will determine the logic and mechanisms of biological processes and the consequent diseases when those processes are defective, made defective in some way. Sorry. And so that really gives biological mechanism a basis for what happens in the clinic and connects the research communities in trying to build this continuum, as I discussed. It will lead to evidence-based prevention, diagnosis, therapy, and cures. It will lead to increased worker productivity and higher quality of life as health and healthcare improve. It will prevent and control all types of disease, chronic, infectious, rare, injury, and so forth. And it will democratize health and healthcare. All kinds of populations will be examined, and and the fruits of that labor will be accessible to everyone, and the care will be tailored to individuals. As we learn more, we'll become better at creating clinical trials. They'll be smaller, the cohorts will be smaller, the tests will be faster, and the fraction of successful trials will go up because we can construct cohorts based on what we know about basic science information that focuses the cohorts on smaller groups of patients that are likely to be successfully affected by whatever treatment's being examined. And as we learn more, we'll be better at knowing what drugs actually help people, which ones won't have an effect on, as opposed to those where there will be no effect or an adverse effect. And same with clinical tests. We won't have to give MRIs to everybody who walks in the door. We'll know which patients will be helped, which ones won't. All those things together will end up reducing the slope of the healthcare cost curve. And I actually don't know of other ways to do that. But what we'll gain in this way, making clinical trials faster and cheaper, has a big effect on drug pricing, for example, or it should. It will, in fact, because the payers won't reimburse for drugs that aren't going to help somebody, and so forth. So it reduce the healthcare cost curve. So lots of advantages. It's a big effort, but I think that it's something that promises to have a big impact, and we're really excited to see how it can move forward, and really excited to hear your thoughts and ideas about it in your domain and as it connects to the rest of our enterprise. So I'll stop there and thank you for your attention, and happy to respond to any questions.